Hi, everyone, and welcome to your chapter 11 in your urinalysis course. This chapter will be on semen and semen analysis. Here are the objectives that you should take a look at on your own time, that describe all of the objectives that for this particular lecture. So these are the things that you should be learning about during the lecture and after this particular lecture. So andrology is the branch of medicine that deals with diseases and conditions that are specific to men. Advances in this field of andrology and assisted reproductive technology, ART, as well as increased concern over fertility, especially now that couples are choosing to have children later in life, have led to an increased emphasis on semen analysis on men. Typically, patients that have abnormal results in semen analysis during the analyses that are sent to the clinical laboratory, anything abnormal would most likely be sent to be analyzed in a specialized andrology lab for further testing to determine the need for in vitro fertilization, or IVF. Clinical laboratory personnel are actually also employed in specialized lab like these, like andrology laboratories, and they do perform both routine and specialized testing. Okay, so let's talk about the physiology of the male reproductive system. Their semen in particular, which is part of the male reproductive system, is composed of 4 different fractions that are the testes, the epididymis, the seminal vesicles, and the prostate gland. The bulbourethral glands are also a part of what is called semen. Each of these fractions differs in its composition, of course, and the mixing of all of these fractions, all of these 4 fractions during ejaculation is very important for the production of a normal semen specimen. So we are going to further talk about every single one of these components. So we have the testes, the paired testes. The testes are paired glands found in the scrotum and contain the seminiferous tubules. Okay, so they are located in the lower scrotum, or in the scrotum, I'm sorry. In the lower scrotum temperature, or low scrotum temperature, is optimal for the development of semen. So these seminiferous tubules are there for the secretion of sperm. The Sertoli cells provide support and nutrients for the germ cells as they undergo spermatogenesis. And the sperm mature and are stored in the epididymis, and that's where they develop the flagella. The ejaculatory ducts receive both the sperm from the ductus deferens and fluid from the seminal vesicles. The seminal vesicles produce most of the fluid present in the semen, so about 60 to 70%. And this fluid is a transport medium for the sperm. So the fluid contains high concentration of fructose and flavin, and spermatozoa metabolize the fructose for the energy needed for the flagella to propel them through the female reproductive tract. If there is no fructose, the sperm do not display motility in the semen analysis. So fructose is absolutely necessary for the sperm to be able to be motile. Okay, the prostate gland is a muscular gland. It is located just below the bladder, and it surrounds the upper urethra and helps in propelling the sperm through the urethra by contractions during ejaculation. Approximately 20 to 30% of the semen volume is acidic fluid that is produced by the prostate gland. This milky substance, this milky acidic fluid, contains high concentrations of acid phosphatase, citric acid, zinc, and proteolytic enzymes that are responsible for the coagulation and liquefaction of the semen after ejaculation. Next are the bulbourethral glands. These are located just below the prostate, and they contribute about 5% of the fluid volume in form of a thick alkaline mucus fluid that helps neutralize acidity from the prostate secretions and the vagina. It is important for semen to be alkaline to neutralize the vaginal acidity that is present as a result of normal bacterial vaginal flora. Without this neutralization, the sperm motility would be diminished. So it is important that this liquid, this fluid, this thick mucus alkaline fluid is there to help neutralize the acidity. The acidity from the prostate secretions and also from the vaginal fluids. Okay, so this table right here summarizes the structures of the semen, or what semen is composed of, and what are the structures that are needed for semen production. Okay, so about 5% of the total volume will be the spermatozoa. Seminal fluid is about 60 to 70%. Prostate fluid is about 20 to 30%. And the volume of the bulbourethral glands constitutes about 5% of the total semen volume. And here are the structures and the structures and what is the function of each and every structure that composes the semen. So in the seminiferous tubules of the testes, we have spermatogenesis going on, the production of sperm. And the epididymides, we have sperm maturation. The ducts deferens helps propel the sperm to ejaculatory ducts. The seminal vesicles provide nutrients for sperm and fluid. The prostate gland provides enzymes and proteins for coagulation and liquefaction. And the bulbourethral glands add alkaline mucus to neutralize prostatic acid and vaginal acidity. And here is a picture of the male genitalia. Okay, the top view that you see here is the sagittal view, and the bottom view is the anterior view. Okay, so in here you can see all of the parts of the male genitalia. On the top sagittal view, you would see the bladder, the location of the vas deferens, the urethra, the penis, the glans penis. In the scrotum, you see the epididymides, the testes, and the scrotum, the seminal vesicles, the rectum, ejaculatory ducts, and the prostate gland. In the bottom picture, which could be a little bit hard to see, but you can pretty much see the same thing, but with an anterior view. Okay, so now let's talk about specimen collection for a semen sample or semen specimen. So it is very important that the patient that is collecting the sample on their own, that they understand that we must get a complete specimen. Most of the sperm are contained in the first portion of the ejaculate, making a complete collection essential for accurate testing for both fertility and post-vasectomy specimens. When a part of the first portion of the ejaculate is missing, the sperm count will be decreased. The pH will be falsely increased, and the specimen will not liquefy. Likewise, when a part of the last portion of the ejaculate is missing, the semen volume is decreased, the sperm count is falsely increased, and the pH is falsely decreased. And on top of all of that, the specimen will not clot. So for this, patients have to have a detailed instruction on how to collect this particular specimen. They usually get written instructions beforehand or before specimen collection. So specimens are collected after a period of sexual abstinence of at least 2 days to not more than 7 days. Specimens collected after prolonged abstinence tend to have higher volumes and decreased motility. That's why it's important that the patient knows that there should be a period of abstinence of at least 2 days, but no longer than 7 days. So the World Health Organization, the WHO, recommends that 2 or 3 specimens be collected not less than 7 days or more than 3 weeks apart. When there are 2 abnormal specimens, that should be considered, and this is per WHO, when there are 2 abnormal specimens, that is considered significant. The laboratory usually provides the patient with a warm, sterile glass or a plastic container. The sample is typically collected in a room that is provided by the laboratory, but if that is not, if the lab doesn't have a room, the specimen could be brought from home, but it should be known that the patient has to keep the specimen at room temperature and deliver it to the lab within 1 hour of collection, okay? Also, when you receive the sample in the laboratory professional has to record the patient's name, the date of birth, that is very, very important, the date of birth, the patient's name, and the period of sexual abstinence and the completeness of the specimen. Any difficulties with the collection should be recorded as well. Specimens that are waiting to be analyzed should be kept at 37C. And usually specimens are collected by masturbation into the container. If that is not possible, only non-spermaticidal or non-lubricant containing rubber or polyethylene condoms should be used. Ordinary condoms are not acceptable because they contain spermicides. Okay, so all semen specimens are potential reservoirs for HIV, for herpes viruses, and for hepatitis viruses. So standard precautions must be observed at all times during the analysis and obviously when you receive the specimen. These specimens are discarded in a biohazardous waste, and sterile materials and sterile techniques must be used when we do a culture of the semen specimen, or when the semen specimen is being processed by a bio assay, or when it's being processed, for example, for IUI or IVF. Okay, so what do we do when it comes to a semen analysis? So semen analysis for fertility evaluation consists of all of these factors. So we look at the semen macroscopically and also microscopically, right? There are certain parameters that are reported for semen analysis, and that's gonna include the appearance of it, the volume of how much we get, the viscosity of this particular sample, the pH is going to be analyzed, the concentration of the sperm, and actually the count as well, the motility and the morphology. There are reference values for these, and these can be found in your book, table 11.2. This is page 280, and it shows you, I'll talk about some reference ranges as we move along with every single parameter here, but the volume, viscosity, pH, sperm concentration, count, motility, quality, vitality. Okay, so how much of it is alive, morphology. That is all. There is reference intervals for each and every single one of these parameters that will be reported. Okay, so let's start with the appearance of the sperm. Okay, the normal semen typically has like a great white color. It appears translucent and it typically has a musty odor. Okay, when the sperm count or the sperm concentration is low, the semen appears to be almost clear. If you ever see a semen sample that is very white and turbid looking, that may indicate the presence of white blood cells, and that could also indicate the presence of infection within the reproductive tract. Okay. You may have to order, or you may have to perform a culture when you're doing a semen analysis, depending on the appearance of it, and if you suspect that there's high count of white blood cells. So specimens sometimes can look a little bit red or have like a red coloration to them, and that could be associated with red blood cells, and that is abnormal. When there is a yellow coloration to the sperm, that could be caused by urine contamination, but sometimes that's caused when a sample is collected after prolonged abstinence or with certain medication. Remember that urine is toxic to the sperm, and that does affect the motility or the evaluation of motility. So there must be no contamination with urine when you are, when you are evaluating this sample. Liquefaction is another parameter that is evaluated during when we're evaluating all of the parameters in the semen analysis. A fresh semen specimen is clotted and should liquefy within 30 to 60 minutes after collection. So we needed to record the time of collection when we were evaluating for semen liquefaction. If your sample fails to liquefy within 60 minutes, that could be caused by a deficiency in prostatic enzymes, and that definitely has to be reported. So analysis of the semen actually cannot begin until your sample has liquefied. If 2 hours have passed and your sample is not yet liquefied, you could add something to it like Dobelco's phosphate buffer saline. So you would take equal amount volume of the semen and equal amounts of Dobelco's phosphate buffer saline. Or sometimes what we do is we add proteolytic enzymes such as alpha chymotrypsin or bromelain to induce liquefaction and allow the rest of the analysis to be performed. Okay, so when you do add these treatments, if you have to, because these treatments could affect some biochemical tests, sperm motility, maybe even the morphology, you have to document the addition of these treatments. Okay, let's move on. Okay, now the volume and the viscosity of the sample. So the normal semen sample volume should range between 2 and 5 mLs. You can measure that by pouring your sample into a clean graduated cylinder calibrated in 0.1 ml increments. Increased volumes of semen can be seen after periods of extended abstinence, however. So that's something that you must keep in mind. And decreased volume of the semen sample can be associated with infertility, or it's often associated with infertility, and can indicate also improper functioning of one of the semen producing organs, in particular the seminal vesicles. Also, we have to make sure that we have a complete specimen when we are evaluating the volume, right? So complete ejaculation. Another parameter that is very important when evaluating a semen sample is the viscosity. The viscosity refers to the consistency of the fluid, and it could be related to specimen liquefaction. Specimens that are liquefied incompletely are clumped and are highly viscous. The normal semen specimen should be drawn into a pipette easily and form a small discrete form small discrete droplets that do not appear clumped or stringy when falling by gravity from the pipette. Droplets that form threads longer than 2 centimeters are considered highly viscous, and they can be recorded as abnormal. You can use a rating scale, rating of 0 meaning a watery sample, all the way to a 4, which would mean a gel-like specimen in terms of viscosity. Viscosity can also be reported as low, normal, or high. We must definitely assess viscosity, because increased viscosity and incomplete liquefaction really make the testing for sperm motility and concentration difficult to perform. Okay, the next parameter is evaluating the pH of the sample. The pH of the semen specimen indicates the balance between the pH values from the acidic prostatic secretion and the alkaline seminal vesicle secretion. The pH should definitely be measured within 1 hour of ejaculation, because you want to avoid the loss of CO2 that occurs after 1 hour. The normal pH of the semen is alkaline, and it should range between 7.2 to 8.0. Increased pH could indicate infection within the reproductive tract, and a decreased pH can be associated with increased prostatic fluid. It can be associated with obstruction of the ejaculatory duct or poorly developed seminal vesicles. So the way that we can do this to test for the pH, we can actually take test the semen for pH by using the pH pad of the urinalysis reagent strip. And we can also take the color and compare that with the manufacturer's chart. Okay. All right, now we're moving on to the parameter of sperm concentration and sperm count. So the actual number of sperm present in a semen specimen is very much a valid measurement of fertility. There are various factors that can affect sperm concentration. For example, the number of days of sexual abstinence before the collection, infection, stress, that can affect the sperm concentration as well. So if you're doing fertility studies, typically we analyze more than one specimen, more than one semen sample. So the reference values for sperm concentration are typically greater than 20 to 250 million sperm per milliliter. And the concentrations between 10 and 20 million per milliliter are considered borderline, okay? So you wanna be at greater than 20 to 250 million sperm per milliliter. And borderline would be considered something about 10 to 20 million sperm per milliliter. The total sperm count for the ejaculate can be calculated by multiplying the sperm concentration by the specimen volume. The total sperm counts that are greater than 40 million per ejaculate are considered normal. Okay, so for example, that would be a specimen that gives you 20 million sperm per milliliter times 2 mLs, which is the volume. So you want to take the concentration times the volume, and anything over any total sperm count over 40 million per ejaculate is considered normal. All right, so when we are evaluating a semen sample, the sperm concentration is typically performed using a Neubauer counting chamber. The sperm are counted in the same manner as the cells are counted in the CSF analysis. We could dilute the specimen and we can count the cells in a Neubauer chamber. This is also called a hemocytometer. The amount of the dilution and the number of squares that we can vary among laboratories, and that those are 2 parameters that we have to include in our calculation for our final number. The most common dilution that we'll use for a sperm sample is at 1 to 20. You need a mechanical pipette to make the dilution. And dilutions actually for semen analysis are essential because it immobilizes the sperm before we count. So we need to make sure that that sperm is nice and immobilized. Otherwise, it would be very difficult to count the cells. A typical fluid that we will use for diluting is sodium bicarbonate and formalin, and these again help immobilize and preserve the cells. You can also use saline and distilled water to make those dilutions as well. So typically, when you perform a semen analysis, you place the diluted sample into the 2 chambers of your Neubauer hemocytometer, and you count the 4 corner squares and the center square, the center large square. This is very similar to what we would do if we were to count red blood cells manually in the hematology lab. We do have to do both sides of the hemocytometer. You load both samples, you allow to settle for about 3 to 5 minutes. And those 2 counts between the 2 chambers should agree within 10% of one another. You will then take an average of the 2 counts and you'll put that into your calculations. If your 2 counts you count from one side of the chamber and the other count of the other side of the chamber, if they do not agree with one another, then you would have to do your dilution again and repeat your counts one more time. You use this hemocytometer under bright field microscopy. So your typical microscope that you would use in any lab, in your chemistry or hematology lab. You can use a stain, like a crystal violet stain, to the diluent fluids, and that'll help you visualize the cells a little bit better under bright field microscopy. All right. So what do we do when we count? We count only the fully developed sperm, immature sperm, white blood cells, also known as red or round cells. Those are not included in your typical count, okay? However, because some white blood cells, the presence of white blood cells is significant, you can identify them and count them separately, okay? So stains typically help us differentiate between immature sperm cells or spermatids and also leukocytes, okay? And they can be counted in the same manner as we count mature sperm. A count that is greater than 1 million leukocytes per milliliter is considered abnormal, and that usually comes with inflammation or infection found in the reproductive organs that lead to infertility. Okay, 1 million leukocytes per milliliter of leukocytes, of white blood cells, again, can be associated with inflammation or infection of the reproductive organs that can lead to infertility. The presence of more than 1 million spermatids or immature sperm can indicate disruption of spermatogenesis, and that can be caused by infections as well, such as viral infections or exposure to toxic chemicals or genetic disorders. And here is an example of the Neubauer counting chamber used for your, this is usually used for hematology when we count the red cells and white blood cells manually, but you can use this hemocytometer for your sperm counts or the evaluation of the sperm count and concentration. Okay, so here's how you would go about using that particular formula. And I think I have an example here. Yes, I do. So calculations of sperm concentration really depends on the dilution and the size and the numbers of the squares that you count. Again, we typically count the 4 corner squares and the middle squares. So you count 5 squares in total. When you use the 1 to 20 dilution, typically you will count the 5 squares. So you'll count the large square in the center and then the 4 corner squares. The number that you find, the number of sperm that you count, can be multiplied by 1 million, or you can add 6 zeros to equal the sperm concentration per milliliter. So sperm, just something to know, sperm concentrations are reported in millions per milliliter rather than microliters. So that's something to just keep in mind. Yeah, so this is an example. If we were to use a 1 to 20 dilution, so let's say we have our sample, we made a 1 to 20 dilution of our sample, and an average of 60 sperm are counted in five counting squares on both sides of the hemocytometer. So you then calculate the sperm concentration per milliliter, and the total sperm count in a specimen with a volume of 4 ml. So here, instead of 4, it's 3 ml. So you take the number of sperm counted times 1 million. So that would be 60 times 1 million. It gives you 60 million sperm per milliliters. And then you times that number times the volume, which in this case would be 4. Okay, and so that would give you 180 million sperm per ejaculate. All right, so the book in page 282, your chapter 11 has 2 different examples. One using a 1 to 20 dilution with 60 sperm counted, and then one using a 1 to 20 dilution with 600 sperm counted. Okay. So this is the typical method. This is the manual method of counting and the sperm and looking at the sperm concentration. We also use something called disposable counting chambers, and these don't typically need a dilution. Okay. The new power method is still kind of like the standard method that is always being used. But you know, the validity of these methods and counting chambers is also, you know, questioned. So there is some instrumentation that it has been developed and that is available, especially, you know, when it's used in high volumes, such in fertility clinics. Okay, so the next parameter that we're going to be looking at is the sperm motility, right? The sperm motility. So the ability of the sperm, or that the sperm have to be able to move forward. They need that forward progressive movement for fertility to occur. It's very, very important for fertility because once it is presented to the cervix, the sperm propels, like it needs to propel itself or themselves through the cervical mucosa to the uterus, the fallopian tube, and the ovum. So they have a long trip and a long ways to go. So they need the forward progressive movement, and that is critical for fertility. So when we look in the laboratory, when we look at the motility of it, we have to evaluate that by examining an undiluted specimen, and we have to determine the percentage of motility of the cells and also the quality of motility. Okay, so you will use a well-mixed sample. It has to be liquefied within 1 hour of specimen collection. You can also perform this test by doing it at timed intervals over an extended period of time. You can look at the motility of it in a drop of specimen on a microscope slide. Under the microscope, you would put a cover slip on top of it. And then you look at it under the microscope and you make sure that you allow 1 minute for those cells to settle, and then you evaluate the motility. Since this is a manual method of evaluating the sample, this procedure is typically performed in duplicate just to make sure that we have better results, or the results are more accurate. So what we report is the percentage of the cells showing actual forward movement. And we can get that result by evaluating 20 high-power fields. Okay, an alternative procedure is to examine 200 cells per slide and count the percentages of the different motile categories using a manual cell counter. So you will evaluate the cells for both the speed of motility and the direction of motility. And you can use a grading scale of 0 to 4, with 4 indicating rapid motility or a straight line movement, and 0 indicating no movement at all. So the minimum motility of 50% with a rating of 2 after 1 hour is considered normal. So the reference value here for motility would be a minimum of 50% motility with a rating of 2.0 or 2 after 1 hour of collection, and that is considered normal. The WHO uses a, wait, let me move this here. The WHO uses a different scale of a rating of A, B, C, and D. And it does state that interpretation has to be done within an hour. 50% or more of the cells should be motile in categories A, B, and C, or 25% or more should show progressive motility. There is an actual manual for this provided by the WHO, and that's the WHO Laboratory Manual for the Examination and Processing of the Human Semen. That was released in 2010, and that recommends a simpler system for grading motility that doesn't quite include speed, because it is very difficult to standardize the reporting of speed. So this manual grades the motility as progressive motility, PM, non-progressive motility, NP, and immotility, which is IM. So motility must be specified as a total motility, which is PM and NP, or progressive motility. And your book actually has a table, that's table 11.4, that shows you the different criteria as described by the WHO of motility. So you are not you're evaluating the way that it moves, basically, what directions. Okay, now we move on to the morphology of it. So we can't just look at the number of cells, because also the morphology of it can be an issue in terms of infertility. So the presence of these cells that are morphologically incapable of fertilization can also result in infertility. So the evaluation of morphology with respect to the structure, for example, of like the head, the neck piece, the mid piece, and the tail, that's very important to indicate the issues that the person could be having. So for example, there are abnormalities that relate to the head morphology that's associated with poor ovum penetration. And if there are abnormalities in the neck piece or the mid piece of the cells, that can affect the motility of the cell. Okay. So here is the typical morphology of it. You also have a picture of it in your book, figure 11.3, that is normal. So you'll see that the normal sperm has an oval-shaped head that is approximately 5 micrometers long and 3 micrometers wide, with a long flagellate tail, approximately about 45 micrometers long. So here you have the head, which has the acrosome, the cell membrane, and the nucleus. You have the midpiece that has the mitochondria in it for energy, for motility energy, and then the tail. Okay, so let's go through all these parts right here. You have in the head, you have the acrosomal cap, which is a cap, a part of the head that is critical to ovum penetration, and it is enzyme containing. So it is located at the tip of the head. This encompasses approximately half of the head and covers approximately 2/3 of the sperm nucleus. You have the neck piece that attaches to the head, and the tail and the mid piece. The mid piece is approximately 7 micrometers long, and it is the thickest part of the tail because it is surrounded by the mitochondrial sheath that produces the energy that is required by the tail for motility. Okay, so the morphology of that is evaluated in a smear. You can evaluate it in a stained sample under oil immersion. So how you'll make a smear is you'll take about 10 microliters of these specimen. You'll place them near the frosted end of a clean slide, a microscope slide. Then you'll place a second slide with a clean, smooth edge in front of the semen, drop at a 45-degree angle, and draw a slide back to the edge of the drop of semen. You will allow this sample to spread across the end of the slide. When the sample is evenly distributed across the spreader slide, you would lightly pull the spreader slide forward with a continuous movement across the first slide to produce a smear. This is when we typically make a blood smear as well, as using pretty much the same technique. You can actually stain this. You can use Wright stain or Giemsa or Shore or Papa Nicolau stains. It just depends on the kind of preference or whatever the laboratory employs. You will then air dry the slides, and those will be stable for about 24 hours. In order for you to evaluate the morphology, you should evaluate at least 200 sperm, and the percentage of the abnormal sperm is reported. You will report abnormalities in the head structure, and that could include abnormalities in the head such as double heads, giant and amorphous heads, pinheads, taper heads, or constricted heads. Other parameters are going of the morphology are going to include measuring the head, the neck, and the tail size, also the acrosome size. You will evaluate for the presence of vacuoles as well. We will also evaluate the tail of it. Okay, so the tail could be a double tail, a coil tail. The cell can also be a spermatid, which is an immature. And here's in a little bit, I'll show you some examples of what the different morphology that you can look at, that you will be finding. So looking at all of these parameters, it's referred to as the Kruger's strict criteria. Okay, this is a strict criteria evaluation that requires the use of a stage micrometer or morphometry. So right now, the evaluation of the sperm morphology using this strict criteria is not performed routinely in a clinical lab, but it is recommended by the WHO. So it is used in evaluations, well, it's used as part of integral evaluations in specialized laboratories. So normal values for sperm morphology will depend on the evaluation method that is used, but it can vary from greater than 30% normal forms when using routine criteria, or greater than 14% normal forms when using the strict criteria. And here are the different morphologies. You can see abnormalities with the heads, with the normal heads, the double heads, the giant heads, the amorphous heads, the pinheads, also abnormalities with the tails, the double tails, the coiled tails, also the constricted heads and the tapered heads as well, and also the evaluation of immature sperm, which is spermatid. Oh, here's some more pictures of it. You will see the image on the left shows you cells that have double heads, and then the image on the right shows you cells that have double tails. Okay, so when we are looking at these cells, we also have to consider what are round cells. So these are immature cells and leukocytes. So these are considered to be round cells. And you could differentiate this from the normal cells or the normal sperm. And you must do a differentiation when you're doing your count, especially during your morphology examination. You have this peroxidase positive granulocytes that are the predominant form of leukocytes that you'll see in a semen sample. And that can be further differentiated from spermatogenic cells and lymphocytes by using a peroxidase stain. So you'll count the number of spermatids or leukocytes that are seen in conjunction with 100 mature sperm, and the amount that you see per milliliter can be calculated using this particular formula, where N is the number of spermatids or neutrophils counted per 100 mature sperm. S is the number of sperm concentration in million per milliliters. So that is the count. You have N, which is the number of spermatids or leukocytes, and then you have S, which is the actual sperm. Okay. Let's talk about sperm vitality. That's another test that we will use as one of the parameters when we're evaluating sperm. Decreased sperm vitality can be suspected when a specimen has a normal concentration with markedly decreased motility. This parameter, this vitality, has to be assessed within 1 hour. And it is evaluated by mixing the sample with a stain called the eosin nigrosin stain. It is normal to have about 75% living. Again, that vitality measurement or the assessment of vitality corresponds to the motility of it. You can also do seminal fluid fructose test for the vitality. And sometimes the low concentration can be caused by the lack of support medium that is produced in the seminal vesicles. And that is indicated by fructose levels that could be low or absent in a particular sample. So these low fructose levels can be caused by abnormalities of the seminal vesicles, bilateral congenital absence of the vas deferens, obstruction of the ejaculatory duct, partial retrograde ejaculation, and androgen deficiencies. You could screen for these fructose, for the presence of fructose using a resorcinol test that produces an orange color when fructose is present. And this is exactly what I just talked about, the seminal fluid fructose and the test that we can do. A normal quantitative level of fructose is equal to or greater than 13 micromoles per ejaculate. Again, we can use spectrophotometric methods to determine this, and specimens for fructose levels should be tested within 2 hours of collection or frozen to prevent fructolysis. Sometimes there are anti-sperm antibodies that get produced. These can be present in both men and women, and they can be detected in the semen or the cervical mucosa or serum, and they could be causes of infertility as well. It is not unusual for both partners to demonstrate these antibodies, although the male anti-sperm antibodies are encountered more commonly. Okay, so when we're looking at these antibodies, again, they can be present in both men and women, but more commonly they are seen in men, especially after surgery or post or a vasectomy reversal. Sperm typically don't encounter the immune system, so the body will, if present, they will consider them foreign, and the body will automatically have like an immune response to them and create antibodies. Typically, when you see clumps, you could suspect that some anti-sperm antibodies are present, so you'd have to evaluate that or take that for further testing. You can do testing on a female as well, where you can mix the female serum with sperm and check for agglutination, or you can do immunoassays on males. There are two tests that are frequently used to detect the presence of antibody-coated sperm that are mixed with agglutination reactions, and that's MAR test and the immunobead test. The MAR test is a screening procedure that you use primarily to detect the presence of immunoglobulin G or IgG antibodies. The immunobead test is a more specific procedure in that it can be used to detect the presence of IgGs, IgMs, IgA antibodies, and it demonstrates the area of the sperm, which part, either the head, the neck, the midpiece, or the tail, that are being affected by the autoantibodies. Head-directed antibodies can interfere with penetration into the cervical mucosa or the ovum, whereas tail-directed antibodies affect the movement through the cervical mucosa. So when you use this immunobead test, the samples are mixed with polyacrylamide beads coated with either anti-IgGs, IgMs, or IgA, and you have to perform a microscopic examination of the cells that shows the beads attached to the sperm in particular areas. So depending on the type of the beads that are used, the test could be reported as IgM tail antibodies or IgG head antibodies, and so forth. The presence of beads on less than 50% of sperm is considered a normal, as defined by the World Health Organization. Okay, so other things that we do other than evaluating for all of those parameters is we can do some microbial tests on the samples, and then we can do analysis for post-vasectomy. The microbial tests that we can do or chemical tests, are very important as well. So the presence of more than 1 million leukocytes per milliliter indicates an infection within the reproductive system. Typically, we're worried about the prostate and the prostate glands. We can do routine aerobic and anaerobic cultures and tests for chlamydia, trachomatis, mycoplasma hominis, and uroplasma urolacticum. Sorry, and those are performed. Those are the organisms that are performed most frequently when we're doing microbial testing on semen specimens. We could also do chemical testing on semen, and that can include the levels of neutral alpha glucosidase, free L-carnitine, glycosylphosphocholene, zinc, citric acid, glutamyl, transpeptidase, and prostatic and acid phosphatase. Just as decreased fructose levels are associated with a lack of seminal fluid, decreased neutral alpha glucosidase, glycosylphosphocholene, and L-carnitine can suggest a disorder of the epididymis. Decreased zinc, citric acid, glutamyl, transpeptidase, and acid phosphatase can indicate a lack of prostatic fluid. Okay, so that's just exactly what I talked about. All of these chemical levels, the neutral alpha glucosidase, zinc levels, citric acid, and glutamyl transpeptidase, and acid phosphatase. Low levels of these can indicate a lack of prostatic fluid. And here are some of the reference semen chemical analysis and the reference intervals for these particular tests. Okay. And one last thing to mention is we want sometimes to detect the semen, so do a post-vasectomy semen analysis on certain samples. So the post-vasectomy semen analysis is a much less involved procedure compared with infertility analysis, because the only concern here is the presence or absence of the sperm. The length of time required for complete sterilization can vary greatly among patients and depends on both the time and the number of ejaculations. Therefore, finding viable sperm in a post-vasectomy patient is not uncommon, and care should be taken not to overlook even a single sperm. Specimens are tested routinely at monthly intervals. So they begin at 2 months post-vasectomy, and they will continue until 2 consecutive monthly specimens show no sperm whatsoever. When we evaluate these, it is recommended that we include the examination of a wet preparation using of a sperm sample using a phase microscopy for the presence of motile and non-motile sperm. So a negative wet preparation followed by a specimen centrifugation for 10 minutes and examination of the sediment is what is recommended. And here, again, the only concern with post-vasectomy is the presence or the absence of the sperm. So remember that only one sperm is required for fertilization. So for post-vasectomy samples, it is very, very, very important that we get that we begin the evaluation at 2 months post-vasectomy, and that we continue until 2 consecutive monthly specimens show no sperm whatsoever. And lastly, the quality control during these the evaluation of semen is as always, subjected to quality control. So it is very, very important that we use control materials when we're doing this overall analysis. And I know that there is some subjectivity that's associated, especially when you evaluate the motility and the morphology of that, because there's really not a lot of control in terms of that, and it's going to be subject to what the person is seeing. But so it is very, very important. It's highly, this is actually rated as a high complexity test under the Clinical Laboratory Improvement Amendment Act, and standards for testing personnel must be observed. So it is very, very important that we stick to the CLIA standards when we are evaluating this, because it is considered high complexity testing. So we have to stick to the CLIA standards and the WHO recommendations when evaluating all of the aspects of sperm. Okay, so that ends your lecture on semen and semen analysis. This is chapter 11 in your urinalysis book. If you have any questions, please let me know. Thank you.